Okay, in this last part of chapter 14, we're going to look at motility in non-muscle based cells, so cells that crawl, generally speaking. So many non-muscle cells are capable of crawling over a substrate, that's just what they're growing on, using lamellipodia and or filopodia. So cell crawling involves distinct events. There's extension of a protrusion, attachment to a substrate, and then generation of tension, which literally pulls the cell forward using the front-anchored protrusions. So to crawl, cells extend these protrusions at the front, or that's usually called the leading edge, and that's a thin sheet of cytoplasm called a lamellipodium. And so here's a lamellipodium, that's a thin sheet of cytoplasm, and then a thin pointed extension from the lamellipodium is called a filopodium. So during what we call normal retrograde flow, microfilaments move towards the rear of the protrusion as it extends. So the microfilaments move towards the rear as the filopodium extends. So this retrograde flow results from actin assembly at the growing tip. So there's assembly at the growing tip here, and this leads to flux back here. And so this is dependent on some proteins you've heard about before, ARP2 and 3, and those are those proteins which produce branching, and they also stimulate actin polymerization, particularly in the lamellipodia. So you get these dendritic trees of actin in the lamellipodia. But there's some evidence also that there is a dependence on certain activities of microtubules as well. But we won't worry about that. We'll focus on the microfilaments. So like we say, attachment or adhesion is the technically correct term of the cell to the substrate. That's just the material on which the cell is crawling, is absolutely necessary for cell crawling. So here's Eddie. It's very distracting. New sites of attachment are formed at the front of the cell, and then contacts at the rear must be broken. So this cell on the left here, this leading edge here has got to extend using polymerization and branching of actin, and then this rear structure here that was previously anchored is going to be detached and then lifted up. So the proteins involved in these attachments are called integrins, and so inside the cell the integrins are attached to actin filaments via certain whole bunch of linker proteins, and then the integrins are attached on the outside of the cell to the substrate, to whatever the cell is crawling on. And that structure where you get this, where you get these filaments of actin and you get the integrin proteins, is called a focal adhesion or a focal adhesion contact. And there's a lot of cell signaling going on here, both from inside the cell to out and from outside to the inside of the cell. So the cell is putting down these contacts as it reaches out. It's putting down these focal adhesions when it projects out of the lamellipodia. And then there is contraction at the rear of the cell, which detaches and then squeezes forward due to the action of actin and myosin interactions, and that's under the control of the Rho protein, which we've talked about before, and there's also a regulation of myosin 2 at the rear end of the cell there. And so what we've got to do is have this kind of homeostatic, this homeostasis of old interactions with the substrate being broken and new interactions being formed. So you could kind of think of it as more of like a rolling kind of model, where the front edge is pushed out by the actin polymerization and branching, and the back end is kind of scooched up, and the old contacts are broken and new contacts are made at the front. So the material from the back is going to be pushed forward, basically. So that's one mechanism by which cells crawl. Now, in tissues, cells which are motile will tend to crawl towards or away from certain chemicals or certain molecules that have been released by other cells in the tissues. So, for example, we're here looking at a site of injury in some tissue, and these molecules that are being released at the site of injury, they may be pro-inflammatory compounds, and they may be chemoattractants, and so they may be attracting cells like circulating neutrophils and macrophages. And so the macrophages and the neutrophils, once they sense this concentration of chemoattractants, they can attach to the endothelial cells in a capillary, and then they can extravasate and crawl out of the vessel and into the tissue. So this process where a cell can directionally migrate is called chemotaxis. So directional migration occurs through the formation of protrusions predominantly on one side of a cell. And so that means that the chemoattractant is causing the melopodia and filopodia to form on one side of the cell versus on another side of the cell. So there are lots of examples of diffusible molecules that can act as these cues for directional migration. And this is called chemotaxis. And chemotaxis can be positive, that's towards a molecule, or it can be negative, and that's away from a signaling molecule. And obviously this involves numerous changes in the cytoskeleton and a significant amount of signaling, and we could go back and talk about RORAC and CDC42 in terms of reorganizing the cytoskeleton in response to things like chemoattractants. So that's just a little bit about chemotaxis, and if you take microbiology you'll see a lot more about chemotaxis because bacteria do it extensively. Last thing to talk about here is something called amoeboid movement. So amoeba and white blood cells, for example, they exhibit this kind of crawling called amoeboid movement, and that's associated with the projection of pseudopodia, and this comes from the Latin or the Greek, I can never remember, meaning false foot, pseudo meaning false, and podia meaning coming from pod meaning foot. And so this involves two processes, one process called gelation and one called solation. So gelation occurs as the pseudopodium is extended, and is what happens is more material streams forward. So as if these were pseudopodia, as more the pseudopodia extend, material streams forward and starts to congeal at the tips or along the leading edge of the lamellipodia or at the front of the pseudopodia. And then solation occurs at the rear of the cell, so that will be occurring back here. And so back here the cytosol changes to a more fluid state, and that means it can stream forward. Because you've got to bear in mind, we can keep putting out these pseudopodia, but we've got to bring this material up from the rear, as it were, so the cell can keep moving forward. So we've got contacts up here, contacts back here. We've got to change the viscosity of the cytosol back here, remodel the cytoskeleton back here, and we've got to break those focal adhesions so that we can move all this material up. And up here we're building new focal adhesions, and we're reorganizing the cytoskeleton. So that's a little bit about amoeboid movement, not getting into too much detail on that. That wraps us up for chapter 14. And so that's a pretty neat chapter. I particularly like the material on muscle contraction, so make sure you pay good attention to that. And I like that because it synthesizes a lot of the things we've talked about already. So I'll end there. I'll see you next time when we've got some new material. Bye for now.